This video is Chapter 8 Notes, Part 1, an introduction to mitosis, specifically focusing on the stages of mitosis. Chapter 8 is all about cell division. Cells will divide when they are given a signal to do so, and cells will divide in various circumstances. So one example of cell division would be like an injury. If you got a paper cut on your finger, your skin cells would need to replenish those that are separated to fill in the gap. So that would be an example of mitosis, making more skin cells to repair that injury. Similarly, when you were born, you obviously were not as giant as you are right now. At one point, you were even only one cell big before you were born. So that one cell underwent a whole lot of mitosis until you were a newborn, at which point you continued to do mitosis to grow into the size human that you are today. So mitosis is used for repair, for growth. Also, some organisms use mitosis for reproduction. More on that in a moment. Meiosis, on the other hand, is used only for asexual reproduction. And so there are only a few cells in your body that would ever undergo meiosis. And those would be either in your ovary or in your testes, depending on if you're going to end up creating egg or sperm. So only certain cells will undergo meiosis for sexual reproduction, whereas in general, most cells can undergo mitosis, which would make a clone of them. Asexual reproduction is where one parent creates offspring. That offspring is then identical to the parent. Asexual reproduction uses mitosis. And some examples are shown here. This is a microorganism that lives in water. It's called a hydra. But essentially all it does is split off a little piece of itself, and that little piece is then a clone of the parent, and that little piece is then its offspring, so asexual reproduction. In a similar fashion, a starfish, if you break off the arm can regrow the body of the starfish, and the starfish could regrow the arm. So technically, if I snapped an arm off a starfish, I could potentially end up with two starfish. Those two starfish would be genetically identical to each other. Some species of lizards undergo something called parthenogenesis, which is where the female can produce fertilized eggs. Essentially, the eggs are just a clone of her cells, but the males in those species are not required. So the sperm doesn't do anything because the female can reproduce on her own. So generally in those species, there are no males. They're all females because they're all producing clones of themselves, so then all the offspring would be female babies too. One more unique example of asexual reproduction is this shark here. About a decade ago in an aquarium, there was a shark that was all alone. It had been there for years, and it reproduced. And the people at the aquarium, the scientists, the researchers, were very confused. So they hypothesized that maybe the shark had mated out in the wild before it was caught and had stored the sperm in it until eventually, years later, that sperm then fertilized an egg to give that shark a baby. But when they did genetic testing, they found that the baby was actually, interestingly, a clone of the mother. So maybe more species than we even know now use asexual reproduction. Sexual reproduction, a little bit more obvious, but that is done through meiosis. So we're talking about egg and sperm. Each of the egg and the sperm carry half of the DNA. And thus, when they combine, they contain a full set of DNA. In terms of evolution, asexual reproduction is a huge advantage because you are producing offspring that might be better suited to the environment than the parent, or if the environment changes, that offspring might be better suited to that changed environment. So in terms of evolution, sexual reproduction is huge. Create a baby that's different than yourself, the baby might have a better chance of surviving. Both mitosis and meiosis are examples of cell division, reproduction of individual cells. So when we talk about cells dividing, we're talking about reproducing. Those words, reproduction, division, are used pretty much interchangeably. Now a little bit of vocab to get us started. Chromosomes, that's a word that you've definitely heard of before. Chromosomes are generally shown to appear in this like X shape, although the majority of the time they are not in that X shape. The majority of the time they look like just one single leg of that X, or like a little noodle. And that chromosome is made of DNA, so very tightly packed in here is a double helix that's wrapped around proteins. More on DNA coming in the future months, but this is all the genetic material. It's very tightly wrapped up around proteins. It's very organized in these things called chromosomes. We usually see the X shape because we are usually looking at chromosomes during cell division. And during that time, each leg of the X is a photocopy. So during cell division, the DNA gets photocopied. So like this leg of the X would get photocopied to make two identical legs of the X. So these here are identical. They're photocopies of each other. We as humans contain 46 chromosomes. We got 23 from mom, 23 from dad. So like this one from mom, from dad, from mom, from dad. They're organized into 23 pairs based on the similar traits that they code for. So whereas maybe, let's just use a really simple example like eye color. Let's say on chromosome 1, there's eye color from mom, eye color from dad. One might be blue, one might be green, but they are both representative of eye color. So we call them both chromosome number 1, just one copy from mom and one from dad. The cell cycle is a term that refers to the time from when a cell forms to the time that it divides. The cell cycle does not involve the death of the cell. It's all about replication. So during the cell cycle, we have a single cell. It replicates to make two, and then each of those two could go through the cell cycle to make two more, and so on. So the cell cycle is the time from having one cell to the division into two cells. We'll now go through each of the phases of mitosis. First up is interphase. Interphase is the time when the cell is not actively dividing. Interphase is by far the longest phase of the cell cycle, and some of the cells in your body are pretty much permanently in interphase, meaning that they really would not ever divide. There's 4 parts of interphase, as you can see here on the right. G0 is where the cell is most of the time during interphase. It's waiting for a signal to divide. Those signals could be the absence of a neighbor. So like in the paper cut example, the cells can realize they are no longer touching their neighbor skin cell. So therefore, they would get a signal that they are no longer next to their neighbor, and they would divide. Or a signal could be a protein where cells communicate and talk to each other and say that it's time to divide. So that signal could be a protein signal. But regardless, G0 is a waiting period. Some of your cells really will never divide, like the neurons in your spinal cord. If you experience a major spinal injury, it is possible that you would be paralyzed for life meaning that those nerve cells become damaged and those nerve cells do not divide because they are pretty much permanently in G0. So then that recovery would potentially never happen because nerve cells really don't replicate often, if ever. When a cell does decide to divide, it then progresses through the other parts of interphase. So first would be G1. During G1, the cell physically gets bigger in size. It's not as though every time a cell divides, it should create a smaller and smaller cell. Otherwise, we would eventually have cells that were practically non-existent. So that initial cell needs to get bigger so that the resulting cells could be equivalent in size to the starting cell. During the S phase, DNA replicates. DNA replicates because each cell is going to need to be a clone of the original cell. So if I have 46 chromosomes in a skin cell, that 46 would double before division so that each of my resulting cells could each have the same amount that I started with. If that doubling did not occur, then every time the cell divided, I would have less and less DNA until again there were no more chromosomes. So that doubling is required in order for the formation of two cells that are identical to what you started with. And then lastly, in G2, the organelles replicate. Each of the resulting cells is going to need mitochondria, going to potentially need a chloroplast if it's a plant cell, is going to need smooth ER, rough ER, ribosomes, all these organelles we haven't really yet talked about. But same idea, we need to replicate everything so that the resulting cell is identical to what we started with. When you're looking at a cell in interphase, you can see that there's a cell membrane around the outside of the cell. Then we've got the nucleus in the center of the cell, which also has a membrane around it. So during interphase, the cell looks like what you would picture a typical cell looking like, because probably the majority of cells that you've seen images of are in interphase, because interphase is where a cell spends most of its time. After interphase, the cell is on its way to division because it already is yanked out of G0, meaning the cell is getting bigger, the DNA is replicating, we're now replicating the organelles, so the cell is ready to go, it's ready to divide. So now we move into mitosis. Mitosis has a few sub-steps, just like interphase had G0, G1, S, and G2. The process of mitosis has a few phases in it. The first of which is prophase. So we are now fully into mitosis. The cell is dividing. During prophase, I remember the P in prophase makes me think of pop, and what's breaking open is the membrane around the nucleus. During interphase, all of the DNA replicated, so there's now twice as much DNA as normal in this cell. A cell cannot survive with twice as much DNA. So the cell at the end of interphase is either going through mitosis or it'll end up killing itself, because at that point it's got excess DNA, excess organelles, it's too big in size. So during prophase, the membrane around the nucleus here breaks open and the spindle starts to form. The spindle are these ropes here, and these ropes are going to attach to the DNA to pull them around the cell. Those ropes, called the spindle, are made by an organelle called the centriole. So during prophase, I can identify a cell in prophase because to me it looks kind of like a plate of spaghetti. In the center of the nucleus, I now see all of this spaghetti, which is all of the chromosomes that are replicated. Whereas to remind you, in interphase, I don't see as significant of strands. I don't see things that look like strands of DNA. So during prophase, I've got the nucleus breaking open and the spindle is forming. My centriole, as I said on the previous slide, is the organelle that makes that spindle. The spindle are the ropes that pull the chromosomes around the cell. Eventually, that cell is going to have some DNA on one side and some DNA on the other, such that it can split in half. So that DNA has to move to each side, and the spindle is responsible for that. The kinetochore is a protein at the centromere where the spindles attach to the chromosome. So in my replicated chromosome, I have a protein that's holding the legs of those X's together. So this is a photocopy of DNA being held together by a protein called a kinetochore. And then the spindle will attach and pull each of those photocopies to opposite sides of the cell. Metaphase is probably the easiest one to remember. The M in metaphase reminds me to think of the M in middle, and metaphase is very obvious to see because all of the DNA is lined up at the center. The spindle will then pull each leg of the X an opposite way so that we end up with an accumulation of DNA on each side. But during metaphase, the chromosomes are in the middle. We often call the middle the equator of the cell, just like the equator of the Earth. Anaphase, I remember the A in anaphase makes me think of a part. I am now going to have each leg of my X pulled a separate direction, and that is what I see in anaphase. Some are moving each direction. Then is telophase. The T in telophase reminds me of the T in 2. And during that time, I can start to see the cell membrane pinching in half so that the cell will end up then splitting in half. So during telophase, the cell membrane pinches in, and that's when I can officially start to see that there will be 2 cells. And then to conclude is cytokinesis. During cytokinesis, the cell membrane finishes pinching inwards, and thus the cell is completely split in half. During a plant cell division, we start to see that the cell wall forms in the center. Since in a plant cell the cells are square, the cell membrane doesn't pinch in, but rather part of the cell wall forms between the 2 nuclei. At the conclusion of cytokinesis, each cell is then going to be in interphase. So to conclude, there's a diagram of all of the stages. First is interphase. During interphase, the cell is waiting for a signal. As soon as it gets that signal, the cell will get bigger, the DNA will replicate, all of the organelles will replicate. After interphase comes prophase. Prophase is when the nucleus has popped open and the spindle starts to form. Prometaphase we don't talk about in this particular class. But moving on, then during metaphase, all of the DNA is aligned in the center of the cell. And during anaphase, the sister chromatids get separated to opposite ends of the cell. During telophase, the nucleus is going to reform around each of those new nuclei, so the membrane around the nucleus will reform. And then during cytokinesis, the cell membrane finishes pinching in.